Welcome to the fourth in our series of cannabis population studies with the world expert, what I consider the world expert on cannabis population studies. Not too many people doing them, of course, Dr. Stuart Reece. And today we are looking at cannabis, how it damages DNA, hardware, and software. So over to you. Thank you, Stuart.
Thanks, Gary. Thanks for asking me. Yeah, everybody knows what the DNA looks like. It's a spiral helix. But if you think about it, you've got a brain cell, a heart cell, a muscle cell, and a nerve cell, but they're all different. So if the DNA is the same, if the genes are the same in all those different cells, it must be the way those genes are expressed that's different between the cells. In other words, it's not the hardware that's the same. The genes are the same from one cell to the next. What's different is the way the software must be expressing the genes to produce the different appearances of those different cells and the different functions of the cells. So we're talking about the DNA hardware and the software. The hardware is called the genes or the genome, and the software is called the epigenome, which is Latin for upon or across the genes. And these subjects are discussed in great detail in our upcoming book, Epidemiology of Cannabis, which we expect to be out in the next few weeks. So let's look at the DNA, the genome. As we said, we all know what DNA looks like. It's a beautiful spiral helix, and there's lots of beautiful pictures all over. What isn't widely realized is that the cannabis impacts DNA in just 15 minutes. It takes 15 minutes to start to disrupt the synthesis of DNA, RNA, and protein. And these are the molecules or the macromolecules of life. These are the main information carriers. And it's not just THC. If you look here at this list, it's delta 8, delta 9 THC, 11 hydroxy metabolites, cannabidiol, cannabinol, cannabichromene, cannabicyclol. And a similar effect is produced by levitol, which has the structure of the C ring common to all these cannabinoids and their metabolites. In other words, it's due to this nucleus structure, the C ring on the nucleus, which is a aliphatic side chain on a phenol type molecule, which is a known carcinogen.
So just go back to that one, Levitol. That's not a cannabinoid as such. It's
No,
something in common to cannabinoids. Is that what you're saying?
yeah, that's the nucleus. That's the nucleus.
Okay. Okay.
So let's look at DNA breaks, which DNA breaks looks like that schematically. And it can be produced by energy, X-rays, or light, or toxins, or genotoxins, which actually break the DNA and destroy it. So the point here is this is obviously illustrations of computers. Computers have hardware and computers have software. And on your computer won't work if the hardware is damaged or if the software is damaged. But this says that the hardware and the software interact because the hardware controls the software and the software controls the hardware. So it's very much a two-way street here between the genome and the epigenome. And that's what this is, a picture of people meeting. The genome and the epigenome interact. And right back in 1971, 1973, they found that, see, this chromosome has fallen off. This is a dividing cell, and this is an abnormal dividing cell here. And this is a normal cell grown in, this is a lung tissue culture, and this is stimulated by cannabis, and it's grossly abnormal, as you can see the change from that to that. And that's
Okay. So the cannabis is actually doing that damage directly.
yeah,
And it's been known for what, how many decades?
well,
This is like what, 55 years.
that's right. And this is a pre-malignant change. This is a neoplastic change.
Okay.
This is Stancheva 1974, and each of these photomicrographs has got a picture of a broken chromosome, as you can see. Lots and lots of broken chromosomes.
Okay. So that's literally shattering chromosomes.
Yeah, breaking them a bit. This is DNA breaks.
And that's done by cannabis.
Yeah, cannabis exposure. And this is oocytes, which is egg cells, and lymphocytes. Now, obviously, we know that cells divide. Everyone knows that cells divide into two. But here, see this first panel, the division's incomplete, and there's a chromosome still connecting them. And of course, as the cells separate, this is ruptured. And so you get fractured DNA, which is another way to break the DNA. And here's the same thing a little later. And here's a tetraploid division. It's breaking into four. And here's a fragment that's broken off together with this chromosome not breaking here. And these are chromosomes left behind here. And this is a pentaploid is 5 breaks. And you can see this is very disrupted cellular division under the influence of cannabinoid. That's ova. So that's the egg cells and lymphocytes. So that's tomorrow's generation. Now this is mouse sperm. That's the eggs in the female. This is the sperm in the male. And you can see this is a ring chromosome. It's joined end to end. And this is a chain. There's 4 chromosomes there joined together. And this is chromosomal fragments. Again, here's ring, and there's a fragment there. So this is in the sperm. So it's not just cells in general, but these are the germ cells that go into forming the next generation. Now, this is a very nice illustration of a comet assay. And what scientists do is they take cells and they dissolve their cell membrane with detergents, and then that leaves the nuclear material like that, and they put it in a gel, and they put it between a positive and a negative pole, and the plus will be drawn to the negative, and the negative will be drawn to the plus. And using that, so the DNA migrates up here in the line of the electrical field, and any broken DNA accumulates in the comet tail. So this is a normal comet assay, and this is the comet assay after cannabidiol and cannabidivarin exposure. And look at all the broken DNA in the comet tail, which is not moving as it should do towards the positive anode here. It's all broken off here, and you can see as it rises, the concentration of these things rises, so the rate, the amount of DNA in the comet tail increases. It's pretty high. You know, it's about 6% there, and it's about 12% there with cannabidiol. So this is a very impressive demonstration of broken DNA.
So we should point out that cannabidiol is seen as the safe drug, and this is the one that's on the supermarket shelves. It's not psychoactive. And yet what you're telling us is that it is just as dangerous as any other cannabinoid, certainly as much as THC, if not more so.
That's right, because it contains the 11-tol nucleus on the C ring. And this is just the abstract from that paper, and it talks about micronucleus development, which is an atomic time bomb in the cell in terms of genetic damage, because the micronucleus shatters, and literally the DNA is blown apart, shattered, and it gets incorporated to the main genome, where it becomes genetic junk, and it basically trashes the genetic code. So this is a powerful driver of things like cancer, mental retardation, and birth defects.
But there are repair mechanisms, Stuart. DNA has its own repair mechanisms, but these are just so destroyed that it just can't get it right.
Yeah, this is overwhelming damage. You know, we could talk about micronuclear physiology, and maybe we will at some point, but the one chromosome gets separated from the main mass because, as you just saw, the 2 pronuclei separate during cell division. And some chromosomes get left behind, or they don't completely separate. So these get broken and busted up. Then they get encapsulated in their own membrane around them, which is called the nuclear membrane. But with the micronucleus, it doesn't have all the proteins, but it collects the mitochondria gather around it, and the mitochondrial activity through superoxide generation, the hyperperoxidation, ruptures the micronuclear membrane, and then the DNA is exposed to the enzymes which chop up DNA normally in the cytoplasm.
Yep.
And also, a lot of the processes of DNA replication and transcription involve cutting DNA normally. So they're cutting and they haven't got enough enzymes. So you're generating literally genetic junk, which then contaminates the main information code in the main DNA after micronuclear nucleoplasmic rupture.
It explains so much.
Yeah, so you're literally letting off genetic atomic bombs. This micronuclease, cannabis is known to be positive in the micronuclease for about 50 years. But the significance wasn't really understood. It's only recently that we've understood that this micronucleus thing is a genetic time bomb, and we'll come to more of that. And this is a comet tail in percent of DNA in cheek cells and liver cells. And as you can see, this is cannabidiol, and it rises pretty high. It's 12% there, 15% there in those. And this is DNA base oxidation, right? We know the DNA code is ATCG. And This is the purines, this is the pyrimidines. It affects both. It's non-discriminatory in terms of which bases it damages.
I'll accept your word on that one.
And this is the micronucleus formation. And look at this is binucleate micronucleate and micronucleus. This is 46%, 53%. These are huge numbers. And I was so impressed with it. These are all different major indices of genotoxicity. So this is the cells aren't growing. This is binucleate micronuclei. This is twin micronuclei. That's micronuclei themselves. That's nuclear buds, where you get a bud developing off the edge of the nucleus. Nucleoplasmic bridges. Remember we saw before that the two chromosomes are joined, the two bunches are joined across. Necrosis is cell death, and apoptosis, which is a different kind of cell death.
Okay, so just tell us what the different colours mean on that graph.
The concentrations are listed there. That's the concentration of thing there. But the point is that these are made like the nuclear buds, the micronuclei, nucleoplasmic bridges, necrosis. These are all measures of aging, as well as being genetic damage. These are seen much more commonly in older cells. So this is nuclear evidence, morphologic evidence that you can see under a microscope of nuclear damage and old nuclei inside these cells. So this is particularly the micronucleus as the engine. See, this is more than 50% of cells. That's devastating.
Okay, and the last one I take is apoptosis. So it might be just a typo. Yep. Okay.
And so here we have the same thing represented in a 3D graph. And, with the rising concentration, we get these very high rates of all these things. This data just fascinated me, so I'm trying to represent it because that table there is so data dense, it's a bit hard to actually read. So I'm trying to illustrate that as in all these different parameters, as the concentration rises, so this is severe nuclear damage that we're seeing. Now, remember before we talked about the micronucleus? All the processes involving the hardware and the software of the genes need energy, virtually all of them. So for that reason, the mitochondria gather around the nucleus to supply the energy that it needs. And here they are gathering around the micronucleus. One of the byproducts of mitochondrial function is superoxide, and through a complex molecular pathway, which is referenced here, that ends up rupturing the micronucleus, which lights the fuse on the genetic atomic bomb, a micronuclear collapse. Now so we've talked about the hardware, the DNA. Let's talk about DNA methylation and the epigenome, the software code that's written on the genes, okay? So the DNA double helix we know. What people don't realize is written on the double helix is a methylation code. An extra carbon gets added, which has the effect of turning genes on and off. So it's a software way of controlling gene expression. And that's why this is one of the big reasons why heart cells, brain cells, muscle cells, and nerve cells are all different, because of the software coding. So the DNA, the helix, carries its own software. There are actually many levels of software regulation in the cell, but DNA methylation is one of the best studied. So we'll talk about that because of the interest of time. Now, I'm not going to argue the case that cannabis affects the epigenome. It just does. Amongst scientists, this is a given. This is not debatable. It's not negotiable. It's not up for argument. It's just accepted. And here's a search of PubMed, the National Library of Medicine, the US, performed on the 27th of January, 209 hits linking cannabinoids and the epigenome. So we just take that as a given. We're not going to discuss it. As I've said, the epigenome works, the DNA methylation works to turn genes on and off like a giant switching machine. And the methylation groups drift on and off. They're controlled by the software programming to turn the genes on and off. And this is from, again, the North Carolina Epigenome-Wide Association study, which we call an EWAS, looking at the top 10 functional annotations of cannabis users compared to control. So you've got brain disorder here, you've got liver lesions, leukemia. This is a leukemic cell here. This is a nerve cell. This is cardiogenesis. This is heart formation. And we saw in the heart video that lots of heart defects are linked with cannabis exposure. This is organismal growth. Agenesis, which is no growth. This is neurodevelopmental disorder and cerebral disorder. So this relates to all the autism which cannabis drives.
So we've seen that study before in the previous presentation, and it is tracking changes in the genes caused by cannabis. And annotations is just another word for changes to, right?
That's right. These are the top 10 processes which are disrupted epigenetically in the software by prenatal cannabis exposure.
That's it.
In cannabis users. And then this is after abstinence. So this is after withdrawal. So this is damage in the testicles, in the seminiferous tubules of the testicles that make the sperm. This is brain damage, encephalopathy. This is learning disorders. This is cognitive impairment, which is learning. This is organism death. This is density of vesicles. That's alveolar macrophages in the lung. Pyramidal neurons, that's in the brain. The formation of the hippocampus, which is in charge of memory and spatial learning, and then area of vessel components. So this is blood vessel formation, which fits with all the cardiovascular anomalies of the great vessels that we talked about on those videos. So both, what this says is that both cannabis intoxication and withdrawal are linked with, this is the negative log p-value, strongly linked with all these major pathways. And this paper here that I put the reference up says that the changes in the cannabinoid receptors in the mRNA in blood happen within just 1 hour. So the paper from 1978, the NAHAS group, said it happened in 15 minutes. And in human blood, they're spotting in the test tube. In human blood, they're spotting it in less than an hour. So these changes are not slow. They happen very quickly. And this thing they're talking about down here, this is the cannabinoid type 2 receptor. And this thing here, DNA methyltransferase type 3A, is one of the main writers of the genetic code, of the epigenetic code. This is and TET3 is one of the main erasers. So the genetic code, the methylation marks, that extra carbon, is put on the DNA, it's put on, it's removed, and these are the two of the main players, and they're disrupted in just an hour by the cannabis. This is what this shows. So there's CB2, there's DNA 3A, that's type 1, and there's TET3, and that's highly significant, and that's TET1, which are the erasers. So this is very important. These are the main writers and the main readers. So it's not just that you're breaking the software, you're breaking the epigenome, you're actually breaking the machinery that controls the epigenome. So you can't fix it because you broke your machinery. Does that make sense, Gary?
And and in some cases in 55 minutes or 4 hours, depending which one it is.
That's right. That's right. And then this also extends male and female germ cells via histone modification. Okay, the DNA is wrapped around histones and it's sort of put to bed, it's put to sleep, it's packaged away, it's put into storage around the histones. So the histones allow access to gene expression by controlling what gets in and what gets out. And this is another major pathway of epigenetic control. And the point is that this is disrupted. This is a major epigenetic mark here for gene activation. the CB2 receptor there, which is controlled by CB2. So the histones are changed as well as the DNA methylation.
All right. So when you're talking CB2, that's a cannabinoid. How would you explain that? Cannabinoid receptor.
Yeah, it's cannabinoid receptor. So, and this is looking at psychosis, and they found in a epigenome-wide association study changes in the immune cells and the mitochondrial function, both of which are known to be influenced by cannabinoids. In first episode psychosis, and of course the mitochondrial damage we know leads to damage to the genome and the epigenome, because they all need the energy from the mitochondria. And so if you damage the mitochondria, you indirectly damage the genome and the epigenome.
Wow.
And the DNA methylation, human ovarian follicle in a concentration-dependent manner. This is the human ovary developing, again, in a concentration-dependent.
And that's cannabis affecting the ovary.
Yeah. And you can see this is upregulation and downregulation. There's thousands of epigenetic hits, thousands of hits involved here. And this is just a table to show that lots of genes are affected. And again, here, lots of genes are affected.
So just going back to those hits, those are changes to the genetic code. Each hit is a change.
The epigenetic code, the software.
Yeah, to the epigenome. I just should get that right, yes.
Right, and this is just showing that these are important genes that are controlled epigenetically, and as the THC rises, the percent methylation changes. The THC directly changes the methylation of these genes. And then the next question is the endocannabinoid signaling in the female reproduction. What are the latest advances? And the short answer here, this is a brilliant paper, is that really everything is affected. Now, what people don't realize, there's a lot of talk about CB1 and CB2 receptors, all these different receptors, but there's actually 7 or 8 or 10 different cannabinoid receptors, depending how you count them. So here's the CB1 and the CB2 receptor, but TRPV1 and G-coupled protein receptor 55 is also involved, and there are many other receptors involved in the cannabinoid signaling, and they affect, this is the female reproductive tract, they affect numerous processes. The other thing we didn't say about mitochondria on the way past was mitochondria actually have their own cannabis receptors on them. Now, remember, the cannabinoids are fat soluble, so they get in the cell membrane, the plasma lemma, but they dissolve straight through and they go through the mitochondria. And the mitochondrion has 2 membranes, an outer membrane and inner membrane. And they actually have all the cannabinoid receptors signaling machinery between the inner and outer mitochondrial membrane. So the mitochondria themselves are directly transducing cannabinoid signals, not just from the cell membrane. It's actually direct transduction occurs there. But anyway, the point is here, that it's affecting all these different things of the female reproduction. So it affects, this is ovulation. So women ovulate once a month, mid-cycle, and the ovum bursts out. But all these processes here, this is the cell where it starts, the oocyte, and it grows, it forms a fluid in the antrums called the antral fluid, and then it ruptures, and that's where the egg comes out of. But all these different processes involved in these steps are all modulated by cannabis and the enzymes which control the endocannabinoid system. The body has a cannabinoid system inside it. Then the ovary, the egg oocyte makes its way down the fallopian tube into the ampulla, from the ampulla down to the isthmus and into the uterus. And all these different steps are controlled by cannabinoid. In particular, there's this is anandamide, there's a gradient here, which controls the movement of the egg through the fallopian tube. And this gradient has to be carefully controlled. If it isn't, then you're going to get this, the oocyte will die, it will implant prematurely, everything won't work. These gradients are critical in female reproduction. Then the ovum comes out of the isthmus of the tube into the main uterine body, and it has to implant and grow into the end. This is the lining of the uterus, which is called the endometrium. And you've got these gradients of all these cannabinoids, which carefully control the implantation process of the oocyte and how fast it grows and how deep it grows. And if you mess this up, then you're going to mess up oocyte implantation.
So cannabis is bound to be interacting
Yeah,
just because of all of those CB1s, CB2s.
yeah, the male and female human reproductive system are very rich in endocannabinoid system and its components. So yeah, so this is humans, rodents, cats. These are all the different functions and all the different proteins and the oocyte, the granulosa, and the theca cells here, and all the different ways that cannabis messes up all these different stages in all the species.
Mm.
So, and they've actually been said to be biomarkers of human female reproduction. These are all the endocannabinoids and all the tissues. And these are the references here. So it's not just one or two researchers. There's a huge literature on all of this. And remember we talked about histones. The DNA is wrapped around the histones, and the histones control the access of the genes, of the transcription machinery to the genes, and that's how they regulate cell growth. So mechanisms of lipid metabolism and uterine receptivity and embryo development, we talked about that. And again, just showing this different paper showing how important these things are, and all the different receptors and all the different pathways. It's not just cannabinoids, it's phospholipids, sphingolipids, cholesterol metabolism, which are all related to the endocannabinoid system, which control all these things. It's a very delicately controlled process. And here we've got mitochondria and cholesterol and all the different interacting parts which make the female reproductive tract work. Now, this is an important paper. It shows non-psychotropic cannabinoids as inhibitors of TET1. Remember we said TET1 is one of the main erasers of the epigenetic code, the DNA methylation. And the non-psychotropic cannabinoids, cannabidiol, cannabinol, and cannabigerol, inhibit the erasing of the DNA methylation code, which is a very important message, because it means it's not just about THC and psychoactive cannabinoids. It's the non-psychoactive cannabidiol, cannabinol, and cannabigerol also mess up not just the epigenome, but the machinery that's primarily responsible for reading, writing, and erasing it. And they found that they do this in two ways. One is by binding the ferrous ion, which is at the active center of TET1, which is iron 2+. So they bind the iron, which is at the catalytic site, which makes it go round, and they mess up the docking in the pocket. So they block the docking in the pocket. Enzymes have a pocket, a catalytic pocket, where these amazing reactions, some of these enzymes will make a reaction go 1,000,000 times faster, because they arrange the reactants in the right way and they distribute the charge properly, and the reactions just go like a steam train, right? Mm-hmm. But they muck up the ferrous ion that's in the pocket, and they muck up the structure of the pocket, and that's how they muck up TET1, which is one of the main enzymes of the DNA methylation machine. Mm-hmm. And so this is cannabis epigenetics, just summarizing all the different cells. This is autism and Purkinje cells, schizophrenia, brain astrocytes, Alzheimer's. This is the dorsolateral prefrontal cortex. That's the front part of the brain that you think with, that makes us most human. And you can see a lot of these papers are references in Nature and Science.
Best.
Yeah, this is Safari here, which is the top genetic database in the world for autism. And this is detailed in our book. And again, brain aging is a major thing that's going on here. Cancer, congenital anomalies, nature, science. So now I want to talk about chromosomes. And this is very important. Chromosomes carry the genetic material. We know about chromosomes because they separate when the cell divides. And these are different, very pretty schematics of what chromosomes look like. Now, the cell has two poles. This is normal cell division, and this is all the components stained. And see here, the green is the microtubules. Now that's polymerized tubulin, and these are the two poles of the cell. And here they're stained orange, and they're the chromosomes that count as stained. And see here, the tubulin is stained there. And the chromosomes appear to slide along the microtubules. They actually don't. They're pulled by a molecular motor called dynin and dynactin that pull it towards the plus ends of the microtubule, which are here near the centrioles. And cannabis disrupts the cannabinoids disrupt all of the key steps. They disrupt the chromosome sticking to the microtubule, the rays of the microspindle. They disrupt the spindle itself. They disrupt the microtubules, the tubulin. They disrupt the motors that pull it, and they disrupt the centrioles. So they disrupt all steps of normal chromosomal segregation. And that's why when we come to epidemiological studies, all the measures of aneuploidy, Down syndrome, Edwards syndrome, Patau syndrome, chromosome 21, 18, and 13, and things like broken chromosomes, deletion 22, they're all showing up as a positive signal because cannabinoids are interfering with all the steps involved. And here we have the mechanism of spindle pole organization in human oocytes, which is very complex. But the take home here is that cannabinoids disrupt it. And this is the oocytes and the centrosomes. And these are some of the main proteins. We talked about dynin and dynactin, which are the motors that pull the chromosomes. This is the North Carolina EWAS study here. And NUMA and SEP, these are some of the key proteins that hold the ends of the chromosomes together at the two poles of the spindle. You see strongly positive signals here. And the kinesin, which pulls it the other way, that's the other motor, 218 hits on kinesin. So these are the main motors that pull the chromosomes along. This is the functional annotations. This is some of the different kinesins, and this is the cancers that they're linked with. Tubulin, there's 106 hits on tubulin. Now, you have to understand, the mitotic spindle is microtubules. That's what the rays are. This is polymerized tubulin, and tub B is beta tubulin. And this is the main alpha tubulin acetyltransferase. This is one of the main enzymes that activates the tubulin, and this is one of the main tubulin isoforms. And it's actually smashing the tubulin, right? So it's like a train that gets derailed. If you blow up the rails, the train's going to come off the rails because you derailed the train. And here we're epigenetically messing with tubulin beta tubulin, which is one of the main components with alpha tubulin, of course, of these dimers which form the microtubules. And this is one of the alpha tubulin acetyltransferase is one of the main enzymes that activates alpha tubulin. So this is diabolical stuff, and this explains the strong signal we get when we look at it epidemiologically. And this is some of the hits from the EWAS study with the p-values. As you can see, they're vanishingly small, 10 to minus 20, even when you correct for multiple study because you've got an EWAS, you've got a lot of hits.
And the smaller, the more significant, right?
Correct. Now let's talk about chromosomal physiology. Remember I said the chromosomes are stuck to the spindle? And that happens they're called centrosomal proteins. So centrosomal proteins. And there are 86 hits in this EWAS study, 86 hits for the proteins that stick the chromosomes onto the spindle. 86 hits, right? And here's some of those hits. And you see the high level statistical significance. These are the centrosomal proteins here, CEP, I, CEP V, CEP K, CEP V, CEP N, CEP X. And these are some of the other ones that perform key functions on the chromosomes. CEP N had so many hits at very high levels of significance. It forms a key function in the kinetochore, which holds the chromosome on the spindle. Now, this is important. The activation of homologous recombination in G1 preserves centromeric integrity. So the chromosome, it's the central part of the chromosome is stuck to the mitotic spindle, okay?
Yep.
That's called the centromere. And this is saying that homologous recombination is central to centromeric integrity. In other words, if there's no integrity at the center of the chromosome, it's going to come unstuck off the spindle, right?
Yeah.
So homologous recombination, that's talking about, this is a mechanism of DNA repair, right? There are many mechanisms of DNA repair. This is high fidelity. This is high fidelity repair.
Yeah. And we've just seen how much damage is being done. So
that's right.
Yeah.
That's right. And what this says, that if you inhibit RAD51, which is the core of homologous recombination, you increase RAD52 and you go to mismatch and joining. What this means is as you block RAD51, which cannabis does epigenetically, Then you go from high fidelity DNA repair to low fidelity DNA repair.
Okay.
Right? And there's the RAD51. There's 9 hits for RAD51. As you can see, there's 9 hits for RAD51 in this EWAS study, epigenome-wide association study, and 1 for RAD52. So you're destroying homologous recombination, which is high fidelity DNA replication and repair. And by default, you're going to low fidelity. And at the same time, you're interfering with the ability of the chromosome to stick on the mitotic spindle, which is why it comes adrift. Right? And so we looked at these in another video. These are the key chromosomal functions: homologous pairing, which is where the 2 chromosomes join up; assembly of chromosomes on the spindle, which happens in cannabis withdrawal; and chromosomal synapsis, which is where they cross over the chromosome from the mother and the father and sexual reproduction, and they cross over, they form synapses, and that's why the kids look like a mixture of the mother and father, because at the chromosomal level they kind of are. But this is where the chromosomes, they sort of kiss and cross over, and the synapse is joined at the molecular level by this machinery, which is disrupted. As you can see, all these genes get disrupted in the thing. So the bottom line is that cannabis damages DNA hardware and software. It directly damages the genome and the epigenome and the maintenance machinery that maintains the epigenome and the genome and the chromosomes. It disrupts the normal mechanism of chromosomal separation, and other parts of the cell, like the mitochondria, which the genome, the epigenome, are dependent on. So you're talking about it's a bit like those missiles that go off; they shatter and they mess it up in lots of different ways. But I do want to emphasize that it not only damages the epigenome, but it damages the machinery that maintains and stabilizes and controls the epigenome.
Okay, well, it's pretty alarming stuff, and it's a mechanism which is driving a whole lot of the population study results that you're obviously addressing in this series. So thanks once again, Stuart.